Thank you. As Jocelyn said, please feel free to stop me also anytime you want if you have any question. I'm going to expand a little bit more about the optical and thermal properties of the cuticle. We all are familiar that plants need light in order to survive, but what we don't usually consider is that plants actually filter light, and that the amount of light that goes inside the cells is not exactly the same amount of light, and definitely not the same quality of light that reaches the surface of the plant. Just a few basic physics. Okay, when the light interacts with an object, let's say a cuticle, for example, but anything, there are only three things that can happen. Light can be reflected. That's the reason why we see objects. Light can go through the object. This is what we call transmission. And the other option is that light can be absorbed by the object. Okay. So what happens when we study the optical properties of the cuticle within the UV and visible light? Here we have a few examples. These two, Agave americana and brassica, are two leaves. And here underneath we have pepper and grape, two fruits. So the first thing we realize is that reflectance, here in red, is usually quite low within the UV region, that is between 200 and almost 400 nanometers. And the maximum reflectance that we have been able to observe is close to a 14%. Although the literature tells that epicuticular waxes, and that here we have an example of what they look like, have been considered to be the major protection against UV light in plants because they are supposed to reflect UV light, we realize that reflection is actually quite low. That doesn't mean that this that waxes, epicuticular waxes, will never cause a very high reflectance, but it's not very common. What we see within the UV region is a very high absorbance. What means is that the cuticle is able to absorb UV light, and that is the main mechanism of photoprotection that we are seeing at the cuticle level. Of course, transmittance, that means the light that goes through the cuticle and enters the cells, is usually very high within the visible light, which makes sense since plants need to carry out photosynthesis. If we compare also how the optical properties of the cuticle change during organ development, here we have an example of tomato. Between very young stages of development and ripe tomatoes, we realize that absorbance goes up. So in very early stages, the cuticle is not such a good photoprotector as it is in later stages, not only in UVC, but also UVB and UVA. And as the absorption increases, the amount of light that is able to go through the cuticle decreases. And of course, we don't see any special pattern in reflectance with development. Okay, as Jocelyn told us, the cuticle is composed of a cutie matrix, here represented in orange, waxes that can be either located on the surface, and I don't know why I'm pointing because I don't seem to find the pointer, that are present on the surface of the cuticle or inside the cuticle, as we can see these black lines, polysaccharides that come from the cell wall, and another component that is very, very important for the optical properties of the cuticle are phenolics. And here we can see on the right which kind of phenolics are present in the cuticles. They are usually phenolic acids, aromatic compounds, as we can see here. And in some species, such as tomato, we also have some flavonoids. What I would like you to see here, go here, why not? And I would like you to see this double bond that is present in red. That is what we call a conjugated double bond. The only thing I want you to realize is that it's present in most of the phenolics that we have found in cuticles. And this double bond, when it's close to the ring, does very important things. So these compounds, what they do is they absorb UV light, okay? But once the light has been absorbed, the question is what happens to it? Well, there are two ways things can go. Once the energy is absorbed, one option is to release it. To release it as fluorescence, as light. But as we can see, in general, cuticles have a very, very low fluorescent emission. Here we see cross-sections of two leaves, and the two leaves and the two fruits, and the amount of the amount of fluorescent light that we are seeing is minimum. Here in pepper, we see something, a very, very tiny bit in agave, but in brassica, that has an extraordinarily thin cuticle, we can barely see fluorescence. Actually, fluorescence is not the main mechanism of photoprotection that has the plant cuticles. And which one is this mechanism? Well, remember that double bond? Well, that is the key. When the molecule absorbs light, usually UV light, this double bond gets weak and is able to move into a different position. To put it simply, you have the ring, you have the chain, and after UV light is absorbed, it moves. Okay? And once the molecule has moved, it goes back to the trans state, to the basic state. That gives an extraordinarily fast, here we describe it, ultra-fast process that is photostable and photodynamic. What do we mean by that? What we mean is that this movement can be done 100 million times per second, so it's ridiculously fast. It's a very effective photoprotective mechanism. The fact that it's photostable means that contrary to our sunscreens that we put on our skins and 2 hours later we need to reapply because the molecule has degraded, this one does not degrade. And photodynamic means that it's continuously moving from one state to the other, releasing as energy as it goes back to the trans state. Okay? Does it make sense? Well, this is how the cuticle interacts with UV and visible light. But the cuticle can also interact with with infrared light. And as we can see here, the cuticle usually has a very, very high absorbance in the infrared region, very low reflectance that we are seeing in blue, and an intermediate transmittance. That means a fair amount of light goes through the cuticle, but not as much. Of course, this is what happens when you have an extraordinary well-developed cuticle, such as is the case of olive fruit or tomato fruit. But when we study leaves, such as an orange, what we are seeing is that the cuticle is very thin. Here we are seeing staining in red. And in this case, the amount of absorption is much lower, and transmittance, going through, is the main mechanism. And which cuticle compounds interact with infrared radiation? Well, actually, every single one of them. Cutin, waxes, polysaccharides, and phenolics, they all can absorb and or reflect infrared light. So, to sum up, what we are saying is that the cuticle interacts with UV light, mainly by absorption, with visible light, mainly through reflection, the reason why we see objects, and with infrared light by a combination of absorption and, to a certain level, reflection. I haven't mentioned near-infrared light. I know it is very important in agriculture. We use it very oftenly in the hyperspectral imaging as a way to determine how much water there is in a crop. And the truth is, we don't know very well how the cuticle interacts with near-infrared light. There are some very old reports that suggest that the cuticle is also reflecting a fair amount of light in this region. And this indicates that when we are seeing our hyperspectral images and we are seeing, okay, this crop needs water, etc., there is a part of it that is the cuticle. So that means that the cuticle is absorbing light, okay? Once it is absorbed, it needs to go out somehow. And how does it do it? Most of the times by thermal emission. So it grabs light, especially UV light that is very energetic, and it's dissipated in every possible direction. Not to the outside, not to the inside, in every possible direction. But this causes a red flag, because what we are suggesting is that the cuticle is being heated up, or not. Because if a plant is under daylight continuously, especially if we think about a very hot summer, it is absorbing a lot of light and is dissipating it into heat that sometimes will remain within the cuticle. So is the cuticle being heated? Well, not exactly. The cuticle can act as a thermal regulator. And here I would like to introduce a concept called the heat capacity. That is how much energy we need to, put it in other words, how much energy an object needs to absorb before it changes its temperature. Let's think, for example, about a ceramic. A ceramic can take a lot of heat and it will remain cool. Well, that is the heat capacity. That is the concept behind. The cuticle has a heat capacity that is above that of atmospheric air. That means that the cuticle is able to act as a heat sink and moderate the amount of thermal energy that goes inside the cells. I'm saying moderate. It will not prevent it. The reason is very simple. The cuticles are not extraordinarily thick, so there is a question of how much material we have. Which cuticle compounds are responsible for this heat capacity? Well, the lipids. The cutin matrix, that is, as Professor Jocelyn mentioned before, and waxes, either epicuticular, present on the surface, or intracuticular. And the most interesting thing is that the heat capacity increases with temperature, because it doesn't matter very much if we have a heat capacity that it will not increase with temperature, because with time, as the temperature increases, the whole thing will become very hot. However, the ability to absorb heat without changing its temperature, for the cuticle, it increases with temperature. Okay. And of course, it also increases during organ growth. Here again, we see differences in tomato. This is the button. And here we have 15 days for post-anthesis to ripe tomatoes. And here we have the value of the heat capacity. If we realize that the highest values correspond to the smallest fruits, to the youngest one, and as the heat capacity decreases, we are reaching the ripe tomatoes. We can also see how that ability to absorb heat increases with temperature between -5 degrees, I think it was the beginning of the, to 50 degrees. And that increase is moderate at early ages of development, but it's very, very high in ripe fruits. So the difference between Immature and mature fruits are 0.5, while at the end of the higher values of temperature, the difference is much lower. Okay. Another property that is important for the thermal characteristics of the cuticle is the glass transition temperature. And what is that? Any polymeric material has the ability to switch between a solid state, something that is rigid, hard, brittle, and a rubbery state, something that is very fluid, very deformable. And that occurs at a certain temperature. The cuticle has a glass transition temperature that changes between minus 30 degrees Celsius to almost 30 degrees Celsius. That is, depending on the species, it can vary a lot. And which cuticle compounds are responsible for that glass transition temperature? Well, waxes, phenolics, and polysaccharides, precisely the components that are responsible for providing mechanical resistance to the cuticle. Those are the ones that make the cuticle remain in a solid state and not move into a very rubbery one. Again, the glass transition temperature changes during development. Here we have a favorite model, tomato, and we can see how during growth, during the early stages, the glass transition temperature is below 0. Since tomatoes cannot grow under those conditions, we can always assume that the cuticle will be, during these stages, in a very flexible and deformable state, and increases dramatically during ripening. One important thing is that in tomato, the cuticle has a more or less fixed amount of phenolics during development, and during this period of ripening, the amount of phenolics increases dramatically. That's a very specific characteristic of tomato. And that increase in the glass transition temperature is due to that amount of phenolics. Again, the role of waxes cannot be very well seen here because we compare the red dots with the blue triangles, the differences are minimum. But that is because tomato, at least the domesticated tomato, has a very, very low amount of waxes. And in order to have a more dramatic effect on the glass transition temperature, you will need a higher amount of them. Finally, the role of polysaccharides can be observed once we remove them from the cuticle, and we see how the glass transition temperature drops dramatically, between 10 degrees and even, in some stages, 25 degrees. Okay. How do we sum this up? Well, from what we know, this is what's happening from a thermal perspective to the cuticle. During early stages, the cuticle is able to absorb more heat without changing its temperature, but at the same time, it is above the glass transition temperature, which means they are in a rubbery state. As Jocelyn mentioned earlier, during this period, the cuticle needs to accommodate a lot of organ growth. And so it makes sense that it will be very flexible, very deformable, and very easy to change its shape. During ripening, on the other hand, the cuticle absorbs less heat without changing its temperature, and they are in a more rigid state. That means they are more mechanically resistant, but at the same time more prone to cracking sometimes. Okay. However, all this information comes from the studies in tomato. We need to study more species. We know what's going on in tomato, but we don't know if this is applicable to what's happening in grapes, apples, etc. Now let's move into the role of environmental conditions on the cuticle. The environment can have a dual effect on cuticle. For one thing is that it changes cuticle biosynthesis during growth. So how much cuticle is synthesized and which components are synthesized will change depending on the environmental conditions. And the second thing is that the cuticle properties will change depending on those environmental conditions. Let's start with the biosynthesis. Okay. The fact that the environment influences cuticle development is something very well known, and it's been studied for many, many years. Here we have an example of a work done in the 70s of the last century, where they already mentioned that in the early 20s, they were already studying the effect of relative humidity in cuticle development. And they realized with very, very basic and simple experiments, such as putting a glass bell covering a plant, they realized that the very high relative humidity had a negative impact on cuticle biosynthesis, much similar to what happens when you do in vitro culture. But there are also some other traits, some other characteristics that affect cuticle biosynthesis. The soil, whether we are having soil or an artificial substrate, it will impact on cuticle. Light intensity and quality, they were already mentioning in the 70s, that will affect cuticle. The photoperiod will affect wax composition. Water stress, how much water the plant is receiving, will impact on the amount of waxes and, most importantly, on its composition. And finally, temperature also affects the cuticle synthesis. Okay, let's start with light. Sorry. We all know that the color of fruits are usually a consequence of compounds such as, again, anthocyanins that belong to the metabolic pathway of phenolic compounds. Phenolic compounds, as we have mentioned before, are present in the cuticle. And it's very well established that several genes within this pathway, such as PAL here, chalcone synthase here underneath, here below, and several genes of this pathway are regulated by light. Also, this part of the metabolic pathway of the cuticle that corresponds to lipids, this is also all these genes, or most of them indeed, are regulated by light. We are not still sure to what extent genes involved in the synthesis of cutin are involved, whether they are involved in or they are regulated by light. Okay. This type of experiment has been done many, many times and is a classic. You take a tree and you compare leaves exposed to the sun, leaves that are on top of the canopy, and you compare them to the leaves that are in the very, very low part of the canopy of the tree. You realize that sun-exposed leaves have a very much thicker cuticle. Here we can see all this grayish this gray region is a cuticle compared to this one in the shade. They are much thinner when they are in the shade, but also, and that's important, they have less amount of cuticle. So it seems obvious that light, how much light the plant receives, alters how the cuticle is synthesized. More recently, things got a little bit more complicated. I'm not going to explain too much of it, but I would like to point out this is a very recent paper by a group from Brazil, when they've realized that when they supplement far red light to the plant, the amount of cuticle gets thicker, which makes no sense because we had already seen that the shade decreased the cuticle. We all know that plants perceive sun and shade by detecting how much red light or far red light they are receiving. But what is the difference? Well, these authors increase the amount of far red light without altering the amount of light that goes, the amount of photosynthetically active radiation, which means the plant is receiving all the visible light plus an extra of far red light, which is absolutely uncommon to occur under normal conditions. What this is suggesting is that the effect of light is far more complex than what we think, and that the cuticle is being regulated by the amount of far red light, by the phytochromes that are, as we all know, responsible for this perception of light. So things are a little bit more complicated than saying more light, more cuticle, less light, less cuticle. Now let's move on into drought. Drought has been studied several times, well, many, as you can see here in this graph. And what they have realized is that every single time a plant is subjected to drought, the amount of waxes increase. But not only the amount of waxes, because having more waxes doesn't necessarily mean anything. The important thing is that increases those type of waxes that are responsible for preventing desiccation. They usually tend to be very long-chain fatty acids. In some reports, they have also observed an increase in cutin accumulation. Again, there is a fair number of genes involved in the synthesis of the cuticle that have been identified to be modified by drought. And what happens with salinity? Another important topic. Well, it happens the same. When plants are subjected to an increase in salt, what happens is they increase the amount and specifically the type of waxes that prevent water loss. Also, there are some reports suggesting that cutin can also be increased. And what about temperature? And I'm not talking high or low temperature, it doesn't matter. Whether it is freezing temperature or 50 degrees Celsius, the plant acts in the similar way, increasing the amount of waxes, because waxes are protecting the structure of the cuticle. So basically, extreme temperature, it doesn't matter both ways, causes the same, an increase in waxes. And although you can probably not see, and it's not the most important thing, most of these genes that we are seeing overexpressed, or for salinity, extreme temperature, and previously for drought, are the same, which indicates that what the plant does is realize there is a stress and act accordingly, touching and implementing always the same genes. Well, these type of experiments are very important, especially if you want to understand the role of a specific stressor, temperature, relative humidity, etc. But this is not what actually happens in the field. In the field, what we're going to have is usually a combination of high temperature with low relative humidity, or in some, depending on the climate, very high relative humidity, and of course, the radiation will also be modified. So what I would like to, and there is also another point that is important. When we see these experiments, there is a question that is very important. For how long was the stress applied? Because if I'm applying a stress, if I'm applying a stress for 24 hours and I'm getting an overexpression of genes and an increase in cuticle, that is fantastic, but that only tells me what's going to happen in the field if during my growing season the plants have one day of very bad temperature or very bad weather, or a week. Something very different would be what happens if I'm growing the plants under those specific conditions from the beginning till the end. So what I would like to show here, some of the results we've been gathering in the lab over the last 20 years in order to understand how the cuticle is modified by the environmental conditions and some cultural practices. Here we have an example. Tomato. We have 3 cultivars of tomato. We compare the amount of cuticle in summer and in winter. And we realize that in winter, and of course, I'm talking about growing tomatoes in winter, not freezing conditions. It's the Mediterranean, so very, very mild conditions. But the effect on the amount of cuticle is tremendous. Check in this cultivar, it's almost 500 micrograms, is very big. And it's not only that there is an effect, but as some questions they have asked previously, there is a cultivar effect. For example, in this case, there is also an increase in summer compared to winter, but it's very mild in comparison to this first one. And here we see, for this case, the aspect of the cuticle in winter and in summer. In summer, obviously, there is much more cuticle, is surrounding the epidermal cells much more. And this increase in cuticle, or this decrease during winter, also affects all the cuticle components, and they affect every single one of them in the same way, proportionately, with the exception of phenolics. Phenolics are extraordinarily affected by the light, especially the radiation, and we see, when we compare summer and winter, a 62% of decrease in the amount of phenolics. Well, in a few slides ago, I mentioned the effect of soil versus artificial substrate. When we started many years ago using artificial substrates, we realized that we need to be very careful when managing irrigation, because it's very easy for the plant to suffer micro-stresses. That, of course, never happens when you're in the soil that has a very, very well buffering capacity. So what happens to the cuticle? If you're not able to manage these micro-stresses, the cuticle will increase dramatically. And it will be, again, in a cultivar-dependent way, because, for example, in this cultivar that has a lot of wild-type genes, the effect is not observed. Another thing that modifies the cuticle is pruning. Leaf pruning is being used very, very often to favor airflow, to decrease relative humidity, and also to decrease the effect of pests such as botrytis. Well, so we developed an experiment to see what happens to the cuticle when we do pruning, because we wanted to know, if we were implementing it in the greenhouse, will it affect the cuticle, all our cuticle analysis? Well, this is what we got. When we do a moderate pruning, when we remove leaves up to the truss that is ripening, there is no effect on the cuticle. We only removing a few leaves, and that nothing happens. But when we do a severe pruning, when we remove leaves up to the truss that is in the mature green state, that means leaving only a few leaves at the top, the cuticle increases very much. And of course, with the cuticle, increases all the cuticle components. And again, since this implies more light and more radiation going to the fruits, there is a much more dramatic increase in phenolics. Another thing that has been used is bags to cover fruits. We all are familiar with them. We know that the use of bags have a negative impact in many cases in the color of fruits, because they can affect negatively the phenolic, flavonoid, anthocyanins that color the fruits. And that depends on the color of the bag. Here you have this, you can see an experiment with grapes where they're testing different types of bags, different colors, transparent, cloth bags, etc. But bags can also generate a microclimate of high relative humidity and even alter the temperature within the bag. Will that affect the cuticle? Well, here we have a work that was done very recently with pears, where we see that the cuticle is reduced during when fruits were bagged. It didn't matter whether it was bagged for a longer or a shorter period of time. The cuticle was negatively affected. So one experiment we carried out in the lab was also to test what happens with these bags in tomato. And the first thing we saw is that, of course, as one can imagine, bagging generated a very high relative humidity condition and produced microcracking. Here we can see 2 tomato cultivars not bagged, and here to the right, those that were bagged. The amount of cracks is tremendous. This doesn't come as a surprise. We all know that water deposited on the surface of a cuticle will induce cracking, especially microcracking. That has been proven in many species, such as pears, mangoes, apples, even sweet cherries, if memory serves. But what happens to the cuticle? Well, in our experience, we were very surprised, because contrary to the previous result that showed that bags had an effect on cuticle thickness, and theoretically, but only theoretically in the amount of cuticle, we didn't see any effect on the amount of cuticle or in its thickness. Here we have the control, 4 different cultivars. And in the pale green, we can see bags that caused an increase in relative humidity, but at the same time allowed light to pass. So a very high relative humidity condition exclusively in the fruit didn't have an effect on the cuticle in tomato, and we will see later on why it happened in pear. However, what we saw is when these bags, besides increasing the relative humidity, reduced dramatically light, the amount of cuticle was significantly reduced. And again, with the phenolic compounds. And again, in a much more dramatic way. See here, for example, that the reducing the amount of cuticle due to the absence of light is moderate in the cuticle, but it's dramatic at the phenolic level. So what's happening here? We are seeing in pears that if you have high relative humidity conditions surrounding the fruit only, you are affecting the cuticle. But here in tomato, apparently nothing happens. Well, there is a very important thing that we need to consider when we compare a pear and a tomato. Pears have stomata, functional stomata, and have lenticels, which are pores that allow gas exchange. Tomatoes do not have that. So most probably, and this is just a working hypothesis, tomatoes are not able to sense that change in the environment, but only to suffer it, contrary to what happened in pears. And now I would like to move on into how the environment affects the cuticle properties. Here we have the diagram, and I would like to focus on the mechanical here on the top, water barrier properties, and thermal properties. Those 3 have been very well studied, and how they interact with each other is well established too. So, the first thing is when we talk about cuticle permeability, we are always considering water going out because it's what bothers us. But when we're talking about the water barrier properties of the cuticle, we also need to consider water going in, because if you alter the barrier, it can go both ways. I like this graph very much because it compares what it is a temperate plant with a plant that is specifically accommodated to grow in under desert conditions. You realize that the transpiration remains constant throughout a range of temperatures between 15 and 35, but once the temperature increases above 35, the water barrier is lost. So water is evaporated very fast. However, in the desert plant, the water barrier increases every time it increases the temperature, but it does so very, very slowly, so that when you reach 50 degrees Celsius, it actually protects its water much better than that of a temperate plant. So, as I said, the water movement across the cuticle depends on the temperature. And unless we are talking about desert plants or plants acclimated to very, very harsh conditions, it will increase with temperature. But it also increases with relative humidity. Here we see the opposite thing, a movement of water inside, and we realize that at 70% of relative humidity, you have approximately 40% of penetration of a liquid after 100 hours. But if you increase the water, the relative humidity to 100%, you get an almost 100% of penetration. Again, high relative humidity decreases the water barrier properties, so allows water to move very freely across the cuticle. And do you remember that glass transition temperature that changes between the very stiff, hard, solid, and a very rubbery-like? The glass transition temperature, that also is modified by the relative humidity and is actually decreased. And how those parameters affect the mechanical properties of the cuticle. Okay, here we go. This is representing the mechanical resistance, the stiffness of the cuticle at the 40% relative humidity here on top with the triangles. We see that the cuticle remains very stiff across a certain range of temperatures, but the moment we reach 30 degrees Celsius, the mechanical resistance of the cuticle decreases. As we increase the relative humidity, it doesn't matter at which temperature, the mechanical resistance of the cuticle will drop. So at an 80% of relative humidity, we still see how once we reach 30 degrees Celsius, the mechanical resistance of the cuticle decreases. However, when we reach 100%, which means when the cuticle is wet, it doesn't matter. The effect of temperature is minimum. And that rigidity is also accompanied by the amount of strength or the amount of force we need to apply to break the cuticle. So, I would like to sum up mentioning that regarding the effect of the environmental conditions on cuticle biosynthesis, extreme temperatures, whether they are very high or very low, affect cuticle biosynthesis, and they tend to increase the amount of cuticle, especially waxes. Drought always increase the amount of cuticle, mainly through waxes. Radiation, especially reduced radiation, have a negative impact on cuticle biosynthesis, although we have seen that things are a little bit more complicated. And relative humidity tends to decrease cuticle synthesis. And what happens to the properties of the cuticle? What happens if I have a plant under very high relative humidity conditions? Well, this is what happens. Under very high relative humidity conditions, this temperature will be lower, and we will have a cuticle that is mechanically weak and will allow water to move through it very, very easily and without much control. If we are under, let's say, a 100% relative humidity, water going out is not such a major problem. But what happens if we have high temperature? Well, actually the same. It mechanically weakens the cuticle becomes mechanically less resistant, but in this case, when the permeability is increased, there will be water loss from the inside. This will be a major issue. That's why we, as a material, relative humidity and temperature are considered plasticizers. That means things that make the cuticle weak. And what happens if we combine high relative humidity and high temperature? Well, up to a limit, there is an additive effect and always a negative effect on the biomechanics. However, when we approach wet, when the cuticle is completely wet, the effect of the relative humidity is so important that temperature does almost nothing. Well, thank you for your attention.